right. Hi, guys. I'm Madav again. I'm with an epidemiologist with the Mississippi Public Health Lab. And I'm going to be talking to you about two pieces of software today, which are really useful, in my opinion, for genomic surveillance. The first is BigBacter, and the second is MicroReact. I think these are both very useful, and they work in tandem really well. So I'm going to start with BigBacter and then kind of move into MicroReact. So, what is BigBacter? BigBacter is a pipeline for genomic surveillance. It can be found at this GitHub address that I've linked. This was developed by Dr. Jared Johnson, who's a bioinformatician at the Washington State Department of Health. And it really combines three main tools. It combines Snippy and Gubbins, but as one tool. It has PopPunk as your second tool, and then it has IQ-Tree2 or RapidNJ as the third tool. And you kind of get four outputs, which are phylogenetic trees, distance matrices, a tabulated summary, which is just kind of the pass-fail sort of details regarding the samples, and a MicroReact output summary file, which is, I think, the most useful thing you get out of it, because the summary file has all of the other three in it. So why is BigBacter helpful? BigBacter creates databases of your samples partitioned into clusters, which are then housed locally wherever you ran the pipeline. And the thing that makes this useful is that every time you have new samples to run, the pipeline compares these samples to all previous samples of that species that you've run and lets you quickly assess new samples. It also creates what are called partitions, which are essentially more closely related samples in each of these clusters as a visualization. They're usually color coded in some way, and it can help identify more closely related samples, even in a given MLST scheme, very easily. Another thing that makes the pipeline useful is you can run it on multiple species at the same time, and then it will separate the species using a sample sheet. And again, this is a very effective tool, I think, for genomic surveillance and visualizing data and communicating that data with a team of epidemiologists so that they can act on it. There are a few limitations, and the first of these, and I think kind of all of these really, is that it relies on a PopPunk database, and I'll talk a little bit more about what that is in a second. But the PopPunk databases are not available for every species. And a list of these databases can be found at this web address I've linked. If the database exists for the species, this is a great pipeline. If it doesn't, it's a little bit of a headache. I think you can build your own database. I haven't tried to do that, but there are instructions that can be found. So what is a PopPunk database? PopPunk is an acronym for Population Using Nucleotide Chimeras. You can find more information at this GitHub address. But essentially, a PopPunk database is a pre-computed file which stores population-level genomic data to enable quick clustering. This allows for rapidly assigning new samples to a known strain or lineage, and the database contains core and accessory genome data, so we can look at both when comparing our samples to the database. But again, you have to have this in order to run it, and you can see that on this next slide. You need Nextflow to run BigBacter, you need Singularity or Docker, and you needed the PopPunk database. You also have to have a few inputs, which are FASTQ files, and you want these to be paired end. If you need a FASTA file, and then you need a sample sheet, which is in a .csv format with 5 columns. You need the sample ID, you need a taxa, you need the path to the FASTA file, and then the path to each of the FASTQ reads. This is an example of the sample sheet just so that if you come back and reference this PowerPoint, you can look at it and see exactly how you need to build it. I just made this up. It's not real. And you can find the full run and install instructions for BigBacter at this GitHub address that I've linked. It should give you a guide on how to download the PopPunk databases for the organisms you need. It should give you an idea of how to install BigBacter. And I believe that it links to instructions for creating PopPunk databases if you need to create your own. Again, I haven't really tried that. And this is kind of what the output looks like for each given cluster. So it should give you a species folder, and then if you click into that, it'll give you the cluster names. And then inside that, this is what you would get. You'd get an alignments folder, the distances, figures, the snippy, some stats about the run, and then some trees. I think the most useful thing here is the figures folder. And the figures folder has a .microreact file in it. Um, and that has the trees, a tabulated summary, and the snippy all included, which I think is very useful and for visualizing. So, what is MicroReact? MicroReact is an open data visualization and genomic epidemiology software. This software was developed by the Center for Genomic Pathogen Surveillance, which is funded by the CDC and WHO and various other government organizations around the globe. And they developed MicroReact in partnership with the European Center for Disease Control and Prevention, or the ECDC. And you can find it at this web address. It looks and functions kind of like Tableau. If any of you have used that, I find it to be pretty useful for visualizing. And so, yeah, why is it useful? It can be used with or without BigBacter. And you can also independently input metadata or Newegg trees, distance matrices, and then use that to generate a variety of visualizations. BigBacter generates a .microreact file for you, and you can upload that file and then modify it by adding other information to fit your needs, which is what I usually do when I'm running this software. This is kind of what the BigBacter output MicroReact file looks like. Should have made it a little bigger, I'm sorry. But if you can kind of see the top left, I've redacted some of this information because there are real samples. But the top left gives you the core genome SNP tree. The bottom left gives you the accessory genome kind of a SNP matrix. The top right has the Newegg tree generated for you as a phylogenetic tree. And you can see the partition metadata blocks and status. It shows you which samples are old and new. And then also the partition section of that shows you which samples are more closely related by color. And this metadata column or box in the bottom right is the tabulated summary. I don't think that's particularly useful. So what I do is once I have this, I rewrite it with my own metadata and kind of reorganize it to be more useful. And this is kind of what that would look like. This is a little bit of a different cluster, but essentially I keep the core genome SNP tree. I think that's the most useful thing for a quick visualization for our epidemiologists, especially in a situation like this where the light colors represent really closely related samples. Then I have a metadata, and you can see that I've reorganized it by feeding it status and partition data. I include our WGS IDs here. The tree you can see now is colored by partition rather than by status. I think that's more useful for a quick glance. And you can reorganize the metadata blocks at the top beside the tree there. So rather than having information about partition or status, I've chosen to include gene data, right? Because I think that's much more interesting and important for epidemiologists. So we can show them, oh, these samples have a OXA23 or 23 like gene. And these, there's no OXA24 or 24 like genes. We can also give them a timeline, which is pretty useful for being able to see where the samples are originating in time, which is very useful for an epidemiologist to track. This is a fake MicroReact file I made without BigBacter just to show you what it would look like if you fed it that data. So again, you could give it a metadata file, which is just an Excel spreadsheet, right? And then you could, if you have a Newegg tree from something else, you can just give it the Newegg tree and the distance matrix from the other pipelines you've run. And you can generate a similar visualization. You don't have to use BigBacter. It's just convenient because it does some of that work for you. But again, here you can see that we've color coded everything by the partitions, by the more closely related samples. We've given our epidemiologists a timeline where they can look and see, oh, these samples in June, for example, are very closely related and populated all at the same time. That might be something worth looking into. It's also important, I think, to change the metadata blocks next to the tree because you can house a lot of useful information there. For example, we can see that most of these samples have OXA23, but the sample 15 there, oh, it has OXA24 and NDM1. Maybe that's something for an epidemiologist to investigate because it's so unique in that. Contrarily, we can say samples 5 through 8 are very closely related. They all have an OXA gene, but also have an NDM gene, and are also all from LTC residents, so long-term care residents. When we're thinking about AMR detection and AMR spread prevention, that's definitely something worth considering, right? Like, oh, a big important gene in a group of patients who are all in a long-term care facility, or samples 11 through 14 very related, different NDM gene, not in a long-term care facility. And so I think this type of information can enable epidemiologists to choose where to, if they need to put their attention first, and how to coordinate a response. And I think that's what makes this tool so useful. You can also choose to include a map if you want, rather than a timeline, or both. But this map is fake. I just picked a bunch of random locations in Mississippi. They aren't real, but you can see the visualization and how epidemiologists might find this type of information very useful. The map can get down to a street level view. You just have to feed it a very exact latitude and longitude. So in summary, BigBacter enables long-term genomic surveillance by creating local databases of samples to run comparisons against for the future. MicroReact is a powerful tool which can create visualizations of the BigBacter data, and the .microreact file produced by BigBacter can and I think should be modified to include more specific metadata for a more useful visualization. MicroReact can also be used independently of BigBacter as long as you feed it the files it needs, so metadata, distance matrices, Newick trees, etc. And finally, I didn't include this in the last part, but you can save the MicroReact file by downloading it as a .microreact file once you've made any changes to keep those changes. I think there are a few best practices to follow that I want to run through very quickly. I think you should always run Phoenix or FLAC AMR or a similar workflow on the samples before BigBacter to ensure that everything passes quality metrics. BigBacter is something you have to run twice. The first time you run it, it'll give you results. You should look at those summaries, and then you can resume the pipeline with the push command to ask it to store the results in your local databases. And so before you store it, make sure everything passes quality. Also, I think you should remove the work directory wherever you're housing this pipeline run. It takes a lot of gigabytes of space. It's very space intensive. I think this is obvious for most of you, but you should never include any patient identifiers in MicroReact. It's housed on a web browser. It's just, and if your state isn't comfortable with you using it as a web browser, I think MicroReact provides accounts and will create specially firewalled accounts for you. You can reach out to them to be in compliance with your state's rules, and it's worth doing. I think you can probably speak a little bit more with Virginia on that. I believe they've done that. I have a few acknowledgments. First, I want to acknowledge Beata and say who worked with me in the core sequencing lab. I'd also like to acknowledge Jared Johnson, who worked really hard on BigBacter. It's a very useful tool, and he's done a great job with it. And I'd also like to acknowledge Alexandra Lawrence with the Virginia Division of Consolidated Laboratory Services, who introduced me to MicroReact and BigBacter in a training she recently provided. And that's everything I have. So if you have any questions, I'll be happy to answer them.
Thank you, Madove. Feel free to use the chat if you have any questions, or you can also unmute yourself. All right. Don't think we have any questions. All right. Well, thank you, Madove. The slides will be available on the Teams page if you want to use this as a resource. Travis, are you ready to go? All
Yep, I'm ready.
right. Well, then handing it over to you.
All right. Well, thanks very much. And Madhav, thanks very much for that presentation. That was super interesting. All right. Now, for the rest of you, you're going to get a little bit of the professor mode today, which means a couple of things is the, scramble mode. And also we're sort of halfway through the training, which means I might be asking you a few questions along the way. There's no tests, but there will be questions. All right, so let's see if I can get this thing launched. And go there. I don't know what I'm sharing. It should be. Can you see the screen too with slides on it?
I see your PowerPoint screen.
Yeah, okay, so I'm going to leave it that way. It's a little strange. I thought I'd get something different, but every day is a little different. Let's see. Sorry, I'm going to bring up the chat so I can see that window. Okay. So ideally, you are seeing the prevalence of risk factors and community-associated carriage of antibiotic resistance, enterobacteria, AC, and AMR genes. So the shorthand that we have for this project is the CRAP project. So it's a community based project where what we're doing is looking at the microbiome of healthy community members who are near Athens or at least commute to Athens on a daily basis. And we're trying to understand their carriage for microbes that have AMR genes in them. And so this is a very big project led by our leader here, Liz Otteson. Aaron Lip is also PI on this grant, but it's a huge project that involved a bunch of students. This was launched slightly unfortunately just before COVID, which meant that we had all the challenges of trying to sample people during the height of COVID. And so what we're trying to do with this is, again, look at what are the AMR genes that are in the healthy population that is around Athens, Georgia, and how can we understand risk factors in that population. So that's really fundamentally the goal. Let's see if I can get this thing to advance. Sweet. Okay, so research questions are, what is the prevalence of the ESBLE genes and CRE carriage in this community? And how effective can we be with metagenomic enrichment is sort of where we're going beyond what the contract was for the initial study, is how effective can we be with the metagenomic enrichment for the AMR genes that we're interested in? And what's the sensitivity if we're doing sort of shotgun metagenomics versus enrichment of those metagenomic libraries, which would tie back to the second talk that we had here for this series. And then third, can we correlate the results that we might see from a more standard qPCR assay from these samples versus what we would get from sequencing? So again, we're trying to sort of tie together what are the different technologies that you've seen presented in this series. All right, so a little bit of background about the study. There were 555 participants, 500 adults, 50 kids. Out of those, we have significant data, which means qPCR, unenriched shotgun libraries with sufficient number of reads, and enriched shotgun libraries with a sufficient number of reads for 267 of those samples. Okay. And so we also have reasonably comprehensive demographic and risk factor data from the metadata for the samples. And again, the samples were collected in Athens, Georgia, but it's not necessarily that every participant lives in Athens. Many of them work here and commute in from nearby communities. All right, so there were a lot of things that have been done with these samples. Okay, so one of the main things was to be able to collect a sample, a fecal sample, then do culture-based enrichment using sort of standard techniques there, then do identification of individual colonies that came out of there, and test a number of individual colonies, typically 50 E. coli, by sensititer resistance profiling. Then we did qPCR on specific AMR genes, 5 of them that I'm going to talk about in a little more detail. We did whole genome shotgun sequencing, or did whole genome sequencing of 1 colony per person. Then we did metagenomic analysis of the shotgun metagenomics of each of the samples. And then we took that same library and enriched it for all of the resistome genes. So everything that you could find in CARD as of several years ago, we made baits to that entire set of genes. And then we've captured that resistome and then sequenced those enriched libraries. Okay, so there's enriched libraries. Unenriched is sort of the first phase of just taking the DNA and turning it into an Illumina library. And then what you want to do with any of the sequenced data sets is take it and map it back to the CARD database to ask which genes have we identified and in what frequency are they there. This RPKM is the reads per kilobase. So that's normalizing for the length of the gene per million reads. So that normalizes for the heterogeneity and the number of reads per sample. And so I'm going to focus on that particular measure. Okay, so if you go back to just the general characterization of these samples, ESBL bacteria were identified for about 4.5% of the population, almost 5% of the adults, about 2% of the kids. All of the isolates were in E. coli, and they were all CTXM beta-lactamases. And we had repeat sampling for most of those individuals that were identified with those ESBL infections. And about 2/3 of those were still present 3 months later. We did not identify any CREs in the 500 samples, which was a little bit surprising. We also looked for risk factors that were associated with that. So ESBL carriers demographically were similar to the overall characteristics of the population that we sampled. So there wasn't sort of one particular risk factor that we could clearly identify. And we had information about healthcare exposure, travel, and a variety of environmental factors. Now, it's also the case that if you only have 25 positives, that's a pretty challenging set of data. So we at least now know how big your sample size would need to be if you wanted to have a more powerful assessment. Okay, again, there's a bunch of sort of specific information about the types of resistance genes that we found, the co-resistance that what those patterns were. We have a variety of information on mobile genetic elements, whether the resistance genes are likely to be on mobile elements, right? So the potential for horizontal gene transfer, there's tons and tons of details that we have from sequencing those individual isolates that we identified with these AMR genes, right? And these are probably all the things that you might expect to see in your own labs and your own data. But one of the things that we have been trying to do as a bonus activity is, can we use the information that we have from this population sample to get a better feeling for how these technologies, these different approaches to characterizing AMR genes, what we would interpret from this population. And in particular, again, what we're going to be doing is looking at qPCR results versus the shotgun metagenomics, so the unenriched libraries. And then we'll take those same libraries and we're going to enrich them, doing hybridization capture of all of the genes that were in card. Okay, we focused on 5 genes in particular because those were ones that we had or could quickly spin up qPCR assays. A lot of these assays you're going to hear about in the next module for looking at wastewater and doing assessments from those. And so these ranged from some genes that were very, very common in this set of samples to some genes that were extremely rare. And so we tried to get a few common, a couple in the 1 or 2 in the middle, and then 1 or 2 that are pretty rare. That was the overall gist. For the sequences, again, this is all Illumina sequence data, paired in 150 reads. Again, the most likely things you're going to see in your own lives. We did QC and trimming of those. Then we did read mapping to the card database. There's a bunch of ways that you can do that. I'm not going to get into the details of all the different ways that you can do that. I'm just going to present results from one of the pipelines that we had. In general, the idea, again, was to take those read counts, the positive hits, use SAM tools and IDX stats to count them and quantify them. Again, we did that RPKM, the reads per kilobase pair per million reads, to normalize among samples and gene size. We filtered for mapping greater than or equal to 10 reads. So these are genes that we had pretty high confidence, quite high confidence that they are present. And then we did comparisons of qPCR versus metagenomic comparisons. All right. I'm going to skip over these in the interest of time and just say, again, we're doing these comparisons that I just outlined. All right. So what did we find? If you compare these different ways of looking at the samples, you can basically summarize this in 3 graphs. One is to look at the enriched scores versus qPCR for these guys in red. And you can see there's a highly significant but sort of modest R score for that correlation. Similarly, if you compare qPCR versus unenriched, you get a similar R score. Again, it's highly significant statistically, but the explanatory power is maybe not really as much as you would like it to be. If you compare the unenriched versus the enriched, the 2 sequence data sets, those are pretty highly correlated. Okay, so you could measure one way or the other and get a pretty good idea of what the other one's going to be. And that's not super surprising. If you then do sort of those same things for all of the different genes that we looked at, ERMB is the one where we have the most data. And so that's a very rich data set where we can try and look for a lot of things. And that we knew was the most common measure. It's in lots and lots of samples. TET-B is the next most common, and we have pretty good correlation with enriched data, but really poor correlation with unenriched. And that's basically because we've sort of traversed this boundary between the unenriched having sufficient count numbers that we have a reasonable number of observations. So we need to do that enrichment to actually get enough counts to start doing those correlations. And then for the genes that are relatively rare that we can't even pick up by qPCR, then you're not going to see anything in those data sets. You're going to need much, much larger sample sizes. Okay, and again, this sort of shows you the idea is that if you have a very common gene, in this case, the ERMBs, those are in all of the samples, right? They're in all the qPCR assays. That's the most sensitive assay. It's in all of the enriched samples, and we detect it in most of the unenriched samples. If you move to something that is less common and therefore less likely to be in high numbers even within a sample, again, you're going to see, we're seeing that about half the individuals are qPCR positive, substantially fewer are detected as positive by enrichment, and even far fewer are detected by unenriched shotgun libraries. Now, one thing, okay, so let me ask this. Here's one of your quiz questions. If you wanted to, could you change where you're seeing these bars for the unenriched ones? And let's just consider TET-B. Is there any way to change the number of samples where you're detecting TET-B? And you can throw it in chat, or you can, volunteer to tell us if you're pretty sure you know the answer. There is a way. Let me start with that. How would you change those numbers? Okay, let me see if I can stimulate a response. Is how much does it cost to sequence 1,000,000 reads on Illumina? Let's go back to the very first. What's a broad number for getting 1,000,000 reads of shotgun data on Illumina? Is it the same for a MySeq versus a NovaSeq? Maybe, maybe not. Okay, so MySeqs, remember, for 1,000,000 reads are about $100 per million. NovaSeq can be down as low as $1 per million. Okay, one way in which you can increase your detection probability is just to sequence more. Okay, so all of these results, even though we've normalized per million reads, if we sequence a bunch more, some of those samples that, are pretty borderline on how many reads we have will jump up to where we expect to see these detections increasing. Okay, now again, by normalizing, we're minimizing that chance. But if you're simply asking, can I detect it at all, increasing the number of reads, which usually means ensuring you're on one of those cheaper, higher throughput platforms, is a way to get to higher detections. You could also say, instead of having a minimum detection level of 10 copies, per million reads, I'm going to make it if I see 5, if I see 3, right? So you can change your sensitivity based on, how risky you're willing to be with what the answer is. It is also the case that the enrichment ratio will change from gene to gene. And even though the enrichment might be working really great for ERMB, because you're starting out with so many samples being positive, you're not going to see that ratio be big. You're only going to see that ratio get pretty good if you're in sort of that, nougaty middle range that tastes really good, where the effective part of your enrichment strategy will work well. Okay, and again, that sort of ties back to a variety of things that Majdi was telling us in the second presentation from the seminar series. All right, so there's lots of public health implications. Again, trying to look into reservoir significance, different methods, different risk assessments. All of these will feed into our ability to provide these pipelines with meaningful answers. Probably the main thing that I really want you to get out of the past, 15, 18 minutes here is that it's difficult to get these pipelines spun up. It's easy to get a measure. It's difficult to start standardizing across measures, and it will take a fair amount of time and energy to bring those sorts of things online. So I think that's kind of the bottom line on that one. If there are no questions, I'm going to move on. I've got another series of slides here where we can have sort of a different point, if that's all right. And I'll let you think about it. All right, so hopefully now you can all see a second series of slides where you're seeing some alligators on the screen.
Yep.
Awesome. Okay, so this is these are slides that summarize a very old study, but it's still relevant to this AMR discussion. And so I want to tie back to Liz's first lecture, presentation in this second set here, where she was telling us, right, that selection pressures can take many different forms and they can have lots of effects. Okay, so here's your next quiz question. In public health, right, and this is one everyone should be able to answer, what are the major messages that we try to communicate to the public about antibiotic resistance? Take the full course of antibiotics. Don't use them when they're not useful. Exactly. Right? So these are, super common public health messages. What I want us to also realize is that everything that we do is in a One Health context, even when it's not super obvious what that context is. Okay, so if I were to ask you, if you're thinking about AMR genes in a One Health context, what are the risk factors for antibiotic resistance that you would immediately think of when you think of tying AMR with also One Health? Okay, which is to say, what are the community risk factors that might be beyond a healthcare environment? Okay, cost is a factor. And where else would we expect in the community to find bugs that harbor AMR genes other than hospitals and other healthcare places. Farms and animal reservoirs, right? So that's super common. Any place else? Colonization of where? Right. So, I mean, the one I immediately think of, and I think many people do, is farms, right? And farm animals getting antibiotics in large amounts, right? So that's pretty common thought. Pets, that's another good one. That's awesome, right? So people oftentimes think about the animals that we're directly interacting with. That's pretty common. Something that's a little less common is for people to think about environmental contaminants. And the one that I want to highlight here are the metals that are released into the environment from burning coal. And one of the major places that we burn coal around the world is to turn coal into electricity. This is relatively old. This is about 20 years ago, where you could find coal-fired electrical power plants in the US. The dot size tells you, you know, how much coal is a proxy for how much coal they're burning. Okay. In 2008, we were burning over 1 billion tons of coal per year in the US. Now we are probably under about half of that because we have converted a lot of our older coal burning plants into solar and wind and other renewable energy. Literally, the total number of power plants is less than half of what it used to be. But what we've done is the small plants have closed, the large plants have stayed open. Okay. And so if you know, think about how these power plants work, the way they turn coal into electricity is they burn the coal, they turn it into steam, you have electricity, and you will generate ash of a large number of varieties, but the two biggest groups are fly ash for the ash that goes up, and you have to capture that, and then the bottom ash that's on the bottom of the boiler. Okay, there's actually all kinds of different ash. It has lots of different characteristics. The big things that I would like you to note is that as little as, 10 years ago, we were producing more than 100 million tons of ash per year. And I want you to compare that to antibiotic use on farms at the same time was around 6 million kilograms per year. Okay, so we're going from a few million kilograms to over 100 million tons. Okay, so the amount that's released into the environment is much greater. We know that this ash is basically, these are metal oxides. And we know that the way that the ash is dealt with is that it goes out away from the power plants, it goes into these settling basins, and then it goes through a series of those basins where the water eventually then is released into a local river typically. So if you compare the metal concentrations for an intake pipe, which would be the open symbols, versus the discharge point for 3 different power plants, 2 are in South Carolina, 1's in Wisconsin, and you could look on a log scale, the amount of metal in that water goes up by approximately an order of magnitude by flowing through that system. And so there have been a variety of studies that have been done over the years on these things. We know that the amount of metal in the water is sort of lowish. The amount that's in the sediment is a little bit higher, but their organisms will bioaccumulate some of those metals, and those can have pretty significant effects on things like tadpoles that might be in those settling basin ponds. And typically, if you have 3 ponds, the first one is largely devoid of life. The second one, you start seeing things in there. And then the third one is a nice marsh that looks pretty good. And so you see those biggest biological effects on sort of that second pond. And so again, we know that the metal concentrations are elevated in these systems. We know that the highest concentrations cause harm to animals, so it's ungood for them to be in those settling basins. If you can keep them out, that's probably a better idea. What we were curious about is, do those same metal concentrations also have any impacts on AMR gene prevalence? And so we did a series of experiments where we took river water that was from the input of a power plant, river water that was at the output into the discharge. So we have a control. We added then additional metal, and then we would add additional antibiotics. And so we could do a direct comparison of pre going into the plant and then after it comes out of the plant. And then we were detecting and looking at what's the proportion of live versus dead bacteria using flow cytometry that were in these aquatic communities. And so we have a couple of publications that came out of that work. Again, this was quite a while ago. And there's a bunch of different results, but I'm just going to highlight one where it's the most clear, right? And not every water system and not every possible way you could make the measures are this clear. This is the clearest one. But if you look at the tolerance at the intake, right? So this is tolerance to heavy metals or tolerance to a variety of different antibiotics. That is always lower versus the tolerance at the discharge. So what that means is the microbial community that makes its way through that power plant and then is released downstream has significant, not only higher tolerance to the metals that they were exposed to, but they were also co-selected for antibiotic resistance, and really a quite broad spectrum of antibiotics, including, frontline antibiotics you would get from a doctor today if you go see them. It is also the case that you can, in mesocosm, so in a very controlled laboratory setting, take water from a variety of sources, increase the metal concentration for a single metal or for a single antibiotic. And if you sort of hit the sweet spot in concentration, you can generate whole communities of microbes, of which you can take them out, plate them, and then characterize individual isolates that have multiple drug resistance, even though they were only exposed to one selective agent. Okay, so, all right, what we've shown is there's a correlation between metal exposure and antibiotic resistance. Then we went into the lab and did these mesocosm experiments so that we showed causality. We know that the doses were environmentally relevant, so we know that that's an actual thing happening in nature. We started characterizing mechanisms, but that's sort of where the team fell apart for other reasons. And we know then there were a variety of mechanisms that were involved, but there's a huge amount of work that can still be done. Okay. And so the thing that I really, want to impress upon a public health audience is, yes, it's true, antibiotic use in humans. It's in us. We are, sort of the most important touch point. It's also true in a One Health perspective, antibiotic use in farms and farm animals and food that we get is also important. But it's also true that there are other human activities that we do on a huge scale that are generating antibiotic resistance genes, novel mutations, providing a selective framework where those are available in the environment then to come into these other systems that we characterize more frequently. So I just want you, again, to be aware that that's the case, accept that that's sort of the reality of the world that we work in. We have parts that we can control more than others. But, taking this holistic approach is pretty important. Okay, and so I hope that's a message that works for you and you find useful. Are there any questions